Hello my dear student, I am Professor Devashish Bose, Head, Department of Criminology and Forensic Science, Dr. Hari Singh Aur Vishwavidyalaya, Sagar, Madhya Pradesh. Today I am going to present a lecture of BSc 3rd semester on the unit Criminalistic, and the topic will be Analytical Samples. This lecture has been jointly prepared by myself and Mr. Giriraj Sharma, a PhD scholar and a UGC JRF at Department of Criminology and Forensic Science, Dr. Hari Singh Aur Vishwavidyalaya, Sagar, Madhya Pradesh. So let's start our discussion while taking a look at what we are going to learn today. Today's lecture is divided into 6 modules. Obviously, Module 1 and Module 6 will be our introduction and conclusion. Module 2 will be advantages and disadvantages of common analytical samples, that is blood, urine, breath, vitreous humor. Module 3, hair, nails, bone and bone marrow, skeletal muscles also. Module 4 will be lungs, brain, adipose tissues, oral fluid, liver, bile and gastric content. Module 5, non-human samples. So my dear student, today we will be discussing on analytical samples. In this lecture, we will be learning about the characteristics, handling and use of specific samples chosen for analysis will be discussed. In the previous lecture, we have learned about the sample handling and their importance in forensic toxicology. Moreover, we also have read almost the antemortem and the postmortem exhibits which are used in the forensic examination. As we know that antemortem samples has their own restrictions such as blood, urine, oral fluid and hair, whereas for postmortem analysis, although any sample may be available after autopsy, the samples most commonly used are viscera. Moreover, here I would like to clear one thing, that in postmortem cases, if the routine sample is not available, then forensic toxicologist can opt for some specific samples, and this specific sample is also known as alternative samples. Advantages and disadvantages of common analytical samples. So my dear students, today we will be discussing the advantages and disadvantages of common analytical samples in the field of forensic toxicology. Common analytical samples in forensic toxicology includes blood, saliva, urine, viscera, hair and oral fluids. We start one by one. We start with blood. As we know that blood as an evidence is one of the most common type of toxicological evidences, both in antemortem and postmortem cases, because everything which we consume gets into the blood and they are distributed. Here we will be discussing about the blood. Blood is made up of 2 parts. One is cellular part and another is plasma part. The cellular part of the blood consists of erythrocytes, leukocytes and platelets, whereas the plasma part of the blood contains organic and inorganic material, and here in this part you will find all kind of drugs, chemicals. At the crime scene, blood could be present in 2 forms, that is wet blood or the dry blood. Collection, preservation, handling and transportation of the exhibit to the forensic science laboratory is important, which will lead to a good result. Following methods should be employed at the crime scene to have the blood as a sample. If it is in a wet condition, then take a swab or use FTA papers, and if the blood is in dry form, we can use the swab. It's a common technique. Cut that area and take scrap, collect entire item, and lifting method for the collection, lift it. So my dear student, after examining the blood, forensic scientist can give his opinion whether it contains the following drug or not, or we move on for the confirmatory examination of the blood, which will finally give out the species, identification, everything, because blood won't go for toxicology, but also for the identification, blood grouping, like A blood group, B blood group, AB or O blood group. These all are important, and finally then it goes to the toxicological part. So this is the function of the forensic science laboratory when they get the blood, not only as a toxicological importance but also for identification. Maybe the things changed. It's not a human blood, or animal blood comes and the sample is something different. So identification, my dear student, go for the identification of blood. Then only go for the toxicological. Apart from all these benefits, blood also has great importance in the forensic toxicology, as I had told you before, for the determination of poison, drugs and chemicals. So let's go through the benefits of the blood in the field of forensic science. It can be easily be made available. From where you want to collect it? It is available. Large database of the blood related evidences, blood related analysis, blood related researches are there. Several collection sites and numerous methods of analysis are available for examination. When we talk about the disadvantages of blood, what are the disadvantages? One thing, it is an invasive technique if the person is alive. But if the person is dead and the body reaches the stage of putrefaction, the stage that time it is difficult to collect sample, and in case of post-mortem sample, they are often hemolyzed or clotted. In addition to this, collection of the drug and poison may be effective after post-mortem, and the site of collection may be influenced by the drug concentration. Moreover, unequal distribution of the analyte between the whole blood and the plasma have a wide variation in interpersonal correlation of the drug concentration with this effect. This is very, and you have to keep this in mind, that the ratio start changing after post-mortem. After post-mortem, if you get the blood immediately, it's good. But after the passage of time, especially after the rigor mortis disappears, it's very difficult. It's a hemolysis, all kind of things happen with the blood. So not a very good sample. And also I would like to mention here that if the person is alive, by force you cannot take blood, either by the order of the honorable court or by some summon, or you can take blood. Otherwise it's very difficult to take blood. No one is going to give blood. Okay, give me your blood and I will test whether you have taken a drug or you are under the influence of alcohol. Under legal implications you can take, but normally it's very difficult. So with this we move on to our next sample, that is urine. It is also an important exhibit with numerous advantages, like it is easily available and also it is available in more quantity. As we know that most of the drugs and their metabolites are detectable in urine at high concentration. Moreover, detection can be done for a longer period in comparison to blood. In addition to this, direct method of analysis such as immunoassay can easily be performed. But when we talk about the disadvantages, such as in post-mortem cases, it is not frequently available. Or if the person is in coma, then you cannot ask him to give me urine. He is alive, but he is in coma. Or he is not in his conscious. So all the urine is lost. Moreover, correlation is an important issue with other biological fluids. It invades the privacy issue, as I have told you before, in the blood. So this is the thing. With urine, this is not the case. Now we move on to another sample, breath. That is breath. We breathe out something. We breathe out. So you can find, suppose you have taken onion, and you can, this person had onion in his lunch or dinner. So this is very important. So my dear student, as you know that breath has been used as an analytical sample for the detection of ethanol in person who are being suspected of driving under its influence. The instrument which is used to detect the alcoholic content in breath is known as breath analyzer or breath alcohol meter, which is a portable device and can be carried out at roadside, anywhere you can take it. In addition to the detection of ethanol in breath, several other drugs including amphetamines, benzodiazepines, cocaines, opiates may also be detected in the aerosol particles contained in the exhaled well for as long as 24 hours after its last use. The detection of these drugs in exhaled breath has become a valuable method for clinical and forensic screening and also for the possible identification of drug abusers. So breath is a very, but again I would like to mention here, it's only possible in ante-mortem cases, means if the person is alive. In case the person is not alive and if the person is in coma, don't try, don't take this. It won't have any legal implications if that person is in coma. Now we move on to another sample, that is vitreous humor, a very new. The beauty of this sample is that less likely than most other samples to be get decomposed or putrefied. Concentration of drug is even in comparison to other samples. It's very good. In addition to this, easy analysis can be performed very easily. When we talk about the disadvantages, what are the disadvantages? It cannot be easily collected, and correlation of drug concentration with blood can be established after the equilibrium is studied. So this is the problem. So let's start with hair. As we know that hair provides a long-term history of drug of use or misuse. If we want to know that whether this person was using drug, hair is the best sample. Moreover, for the determination of chronic poisoning, hair is a very good sample, or even in case of putrefaction or mummification, hair may be used for the determination of poison. Decomposed or putrefied, like the problem does not exist with the hair. Invasion of the privacy, these kinds of issues do not exist with the hair. Moreover, it can be easily collected, preserved and transported for the examination in forensic science laboratory. So here is the latest and very important sample which has been considered these days. Obviously, with the invention of the techniques, analytical techniques which are highly sensitive, like separation techniques coupled with mass spectroscopy. So everything has its own disadvantages. What are the disadvantages? The disadvantages is that very low concentration of the analyte is found. So again, what I have mentioned, with routine instruments it's not useful. Moreover, problems differentiating between exterior and the interior drug concentration, means inside the body and outside the body drug concentration present, analytical and interpretative difficulties are there. But still, it's one of the alternative metrics which are being used. You cannot deny that. Hairs are very important, especially in case of metallic poison. It's a beauty. It's a beauty. Now we move on to another. It's again a dead tissue, nails. As you all know that nails are exoskeletonous and made up of keratin, which is a type of protein. Generally, we know them as dead part of our body and remove them by cutting them from time to time. If you are moving towards the historical aspect of nail, so arsenic may have been the first analyte to have been detected from the nail for the purpose of forensic analysis. After several years, scientists and researchers have also detected methamphetamine, amphetamine as a part of screening, making them to be the first drug of abuse detected in nails. Since then, several other common drugs of abuse including cannabinoids, opiates, cocaines, phencyclidines, benzodiazepines and methadone have been detected in the nails. Commonly, the advantages of fingernail and toenail analytical samples are similar to that of hair. Nails as also grow at somewhat predictable rate of approximately around 1 to 3 mm per month. So we know how it is growing. So we can therefore with their growth we can find the chronic use of drug within them. The determination of nail is especially useful when hair is not available. You can cut the hair, but nails, if it has grown, it's very difficult. You can still scrap out the nails. However, the rate of growth may be influenced by age or other factors, malnutrition, all these things. Now we move on to the next, that is bone and bone marrow. So my dear student, as you all know that bones are an important part of our body. Generally, it is heterogeneous, consisting primarily of two different types of tissues. That is decortical bone, which is dense and compact, and cancellous bone, which is spongy and more porous. If the bones are different, then their distribution of the drugs along the bone is also different, such as the femur that consists of both types of bones. There will be different distribution. Although in forensic science, bone is not commonly collected for analysis, but they are the sample of interest in case of metallic poisons and chronic poisoning of drugs of abuse when hair is not available as a sample, and it is a valuable sample because specific drug may be detected in bone for a prolonged period of time. There are two types of bone marrow, red and yellow. Red marrow is capable of producing hemotropic cells, commonly RBCs, and isolated in the bones of the lower skull, vertebrae, shoulders and pelvic girders, ribs and sternum, whereas yellow marrow consists largely of fat cells and is located in the bones of the hands, feet, leg and the arms, basically long bones if we talk about. Difference in the distribution of drugs in the marrow collected from different bone types, as well as concentration difference in marrow from the same bone type, may be expected due to several factors, including difference in the composition of the type of marrow, age of the subject, postmortem redistribution and postmortem degradation of synthesis. I would like to mention one thing at this point is that bones and bone marrow mostly are basically a postmortem samples. You cannot ask a live sample to give your bone, or we want to take your bone marrow because I am telling you why. Because there are other samples which are available. These samples I am taking, suppose it's a putrefied tissue or you get just a skeleton system. In those conditions, when there is no matrix, you will get nails, you will get hairs, you will get bones. In those cases, bones are important. Gastric content, oral fluid, liver, bile, brain, lungs and kidney. This we are going to study now. So my dear student, in this module we will be discussing about the different samples which can only be collected by a professional medico, that is a doctor, after the autopsy. So after the autopsy, these evidences are of utmost importance and to be sent to forensic science laboratory for analysis. So, or a toxicological lab. So let's start with the discussion of gastric content. These contents are very important. These are very important evidence which sometimes proves to be differential between accidental death or foul play. You can clearly differentiate. Gastric is a beautiful sample. Whenever foodstuff is present in the stomach, it shows what that person has eaten before. That means after the consumption, one thing is very clear, this process of digestion stops as soon as a person dies. So the food material or the things which are inside the stomach will remain as it is. This is in the case when I am saying that a person is dead. When the person is alive, you take out, wash the stomach, just like vomiting, clean the stomach and you will have, look, this is a sample what that person has taken before. So it is also very important for calculating the time since death. Very important sample for calculating the time since death, because foodstuff is digested in stomach at a definite time, a particular time. By observing the change in the state and condition of food, we can estimate the time since death. Stomach content is very useful evidence in toxicological cases because we can examine the drug from the foodstuff collected from the stomach. So my dear student, here I would just want to discuss one example which will show the importance of gastric content. Suppose, imagine in a party, in a midnight party, 4 of the friends went to a dancing bar and have dinner together, had good time, make a merry, and they left differently. After some time, 2 of them started vomiting and died. We don't know, 4 persons went there to a restaurant, dancing bar, dance, ate, drink, everything, and they came back. But 2 persons died. After autopsy, the doctor collected the stomach content and forwarded them to a toxicological laboratory. Forensic expert analyzed the sample and found the presence of alcohol and cocaine in one of the content, and the cause of death was surely due to drug overdose and synergism. That it was due to accident. Accidentally some person took an overdose and it was a synergistic effect. Now in the second person, it shows the presence of barbiturate. So in the another person, it may shows the presence of barbiturate or some insecticide or something which may be given to him intentionally, or he might have taken it for the purpose of committing suicide. So this is the opinion. This is the opinion you can get out of this. I think it is clear, no? What is the importance of this beautiful sample? It's very ugly sample, smelling bad. But if you are looking at a professional way, as a toxicologist or with a toxicology side, it's a beautiful sample. Now we move on to oral fluids. Oral fluids are also very useful forensic evidence. After taking some drug, froth may come out, as you might have seen in the movies, that froth oozing out after taking some poison. That it shows. We cannot, in a movie, you cannot say that someone is drinking something. How you will imagine that what it is? But after drinking, froth coming out. It immediately clicks to our mind that it's a poison, that person has taken a poison. So which should have to be collected by expert? Very important, froth is very important. Forwarded to a toxicological laboratory. FSL and toxicological laboratory. In India, all toxicological labs are with FSL. Whereas in Western world, it's with the medical college or it's with the autopsy center. Here is the autopsy center and next door is their toxicological laboratory. Can you tell what is the benefit of this? The simple benefit is you don't need to preserve nothing. Perform the postmortem, collect the sample, give it to the next door. A toxicologist sitting there analyzing. No delay in giving result. No sample is spoiled, sample is missing, nothing like that. So this is, so I am taking about a middle way, I am talking about toxicological labs. Now we move on to another samples, that is liver and bile. In autopsy sample of liver and bile juice should be collected. Liver is a very good evidence for analysis of drugs. Can you tell me why? Because the biotransformation of drugs are carried out in the liver and the metabolites of that particular drug can be analyzed in the liver. Bile is the digestive juice secreted by the liver and it is stored in the gallbladder. Bile juice also should be forwarded to the laboratory for analysis of drugs or toxicants. Next we move on to another sample, that is brain. Brain and CSF, that is cerebrospinal fluid, are collected in the case of poisoning of or due to the poisoning of CNS or PNS acting drugs, or in those cases where the person had suffered from some neurological disorders. In those cases only it is useful. Next we move on to lungs. Lung sample are very essential for the analysis of gaseous poisons, or we can say volatile poison, or if it has been inhaled in that case. First and foremost, gases which are present in the lung should be collected. Then the sample of the lung should be sent to the laboratory. It should be collected immediately, you know. It would be gone in no time. The gases in the liver, once you take out during autopsy the livers and you fail to collect with a simple squeeze, or when it comes out of the body, it is in one of the enclosures. It's in a cage. So it's still there. But once it comes out, it's gone. So you have to very calculative in collecting this sample after autopsy. Kidney. Kidney is very essential analytical evidence. You know, kidney is the most important sample you have to collect for analysis of drug. All the metabolites of drugs excreted via this route. Kidney. By analysis of analytes, we can identify the drug. Therefore, it is very essential to collect the samples from the kidney. Non-human samples. Insects. My dear students, you all have seen insects like flies, house flies, common other flies, maggots, beetles, etc. Commonly, we consider them as a small creature and think that they do not have importance in our life, forensic importance. However, they are very important in forensic point of view, as when human postmortem samples are not available, means they have been decomposed, they have been eaten away. The branch which is related to the study of insects in forensic point of view is called forensic entomology. Insect samples such as larvae, pupae, adults, pupillate cases collected from cadavers which have been used for the detection of time since death, which is an important part of criminal investigation system, and also it has been used for the detection of drugs. In the beginning of the use of insects in forensic toxicology, most probably phenobarbital was the first substance that were detected in the maggots. That means the person died due to overdose of phenobarbital, the body decomposed, maggots sets in, maggots were taken, and in maggots won't take phenobarbital. They are not drug addicts or druggists. So the maggots basically eaten that dead body, maggots survives on these dead articles, and in those maggots phenobarbital was detected. Subsequently, it has been shown that several drugs including barbiturates, tricyclic antidepressant, opioids, cocaine can be absorbed by the insect feeding on those decomposing bodies. The most desirable insect stage for use of analytical samples are feeding larvae, since the rapid elimination of drug in non-feeding larvae and pupae results in lower drug concentration. Now we move on to another sample, that is soil. Soil is a part of Earth's crust and found everywhere, which is a land part. As you all know that in many rituals after death, the dead body is cremated, so it is completely converted into ash, and then it has less importance except for metallic poison. But in some rituals, bodies are buried in the soil. Many times, offenders dispose of the body by burying it after committing some crime, murdering or something. The soil become now the importance of forensic. That means forensic important evidence is the soil. When it is used in criminal investigation, for example, in case of suspected death, in which death occurred due to the overdose of a drug or some metallic poison that have been detected in the soil beneath the decomposing body of the human. And the experimental animals also have shown very good results for the detection of diazepam, phenobarbital, which were even detected after 2 years' time. Precursors and byproducts associated with the illegal production of methamphetamine also have been detected in the soil. Household and personal samples. So what are these? Households and personal sample and its importance in forensic science. Occasionally, in some cases, related to analysis of household and personal samples like chemicals, aerosol containers, vital prescriptions, medications, syringes, and cigarettes, clothing obtained from scene of crime in cases of homicides, suicide, automobile accidents, or scenes of incidences of other interesting things. Because containers often do not contain the substance which have been listed in the forensic examination, but they are very important when the body disappeared, decomposed, burnt away or something. So these samples are very, very, very important. With this, we come to the end of module 5. After the discussion of different analytical samples in the above given module, we can conclude that all the analytical samples are very essential to prove whether the death was due to accident, suicide, or it was homicide. As well as by using analytical samples, we can estimate the time since death. Keen observations of result of analytical identifies the particular drug or poison. By analysis of blood, we can solve the case of paternity and maternity disputes. As well as blood is very useful evidence in the form of personal sufferings like deadly diseases. Nails and hair samples are very useful in metallic poison cases, while bone and bone marrow is useful in case of drowning. So all samples are very important toxicologically as well as others. Samples of lungs and air are very useful in case of volatile or gaseous poisons. If the person is suffering from neurological disorders, or if that person has consumed some CNS poisons, then the brain cerebrospinal fluid are very important. Therefore, we can say that all analytical samples are working as a boon to solve the poisoning cases. With all these information here, we come to the end of today's informative lecture. Do keep in mind what we have discussed today. So, it's Professor Devashish Bose signing off for today. I will be back with some more lectures and informative things. If you want to enhance your knowledge, you may log on to our website at www.cec.nic.in for more MCQs, quizzes and LORs. Till then, goodbye and thank you.